Good evening, ma'am.
Yeah, good evening. We'll just wait for some time for the students to join and then we'll start with the session.
Sure, ma'am.
Okay, so I guess we'll start with the session. Um, I hope my screen is visible to all of you. Please let me know that the screen is visible.
Yeah, sure, ma'am. Is it visible?
Okay. So, if there are any new students, let me just introduce myself to you. I am Prashasti. I am a PMRF fellow and a PhD scholar in Banaras Hindu University, and I'll be taking this discussion session, which is for the plant developmental biology course that you have been undertaking, whose instructor is Professor Sri Ram Yadav. And these discussion sessions are meant to basically help you solve your assignments that you are getting every week. And we basically here will discuss the previous year's assignment questions, and I'll give you some time to answer me those questions. If you're able to answer, very good. Otherwise, I'll answer the question, and we'll just learn a little bit about that topic on which the question is asked. And this way we'll go ahead with this session, and if there's anything else that you want me to ask or you have any doubt or anything else, please let me know, and we'll now proceed with the session. So, okay, so this is the first question. TOS17, a retrotransposon, is known to get activated at which developmental stage? Callus, during organization of meristem, embryogenesis, or fertilization? I'll give you some time if somebody answers very well, otherwise we'll go ahead by understanding this. Okay, so we'll proceed with understanding this question. So let's first try to understand what retrotransposons are. So retrotransposons are nothing but they are genetic elements that move around in the genome by copying themselves as RNA. So basically, we all know that all the organisms have some kind of genetic material. It can be either RNA or it can be DNA. Now, most of the organisms like humans, like animals, like plants, all of us, we have the main coding genetic material as DNA. And also, because DNA is very much more stable as compared to RNA, it is easy to transfer DNA. But the retrotransposons, they are nothing but those kind of genetic materials which move around or copy themselves as RNA. And then later on, that RNA gets converted back to DNA. So, in certain cases, like in plants and animals, these are found basically in plants or in animals, and they can be mutagenic. That means that the kind of retrotransposon that is being moving around or copying itself in any organism can be mutated or it can be altered with time, and that particular RNA will then get converted back to DNA and then work as a genetic material. So this is what retrotransposon is. Now the question is, how do they work? So basically, retrotransposons use the mechanism of copy and paste. So what happens is, every time there is some DNA which needs to need to move around or something like that, so this DNA will get copied in the form of RNA, and that RNA is mobile. That RNA can move around, and wherever that RNA is destined to move, that RNA will go there and then get converted back into DNA. So this is nothing but copy and paste. The word copy and paste here is used because it is copied from the DNA. It is copied from the DNA in the form of RNA and then again gets pasted in the form of DNA only. So that is why the copy and paste mechanism word is used for the movement of retrotransposons in any genome. Why are we saying this, that the any kind of genetic material needs movement? So movement is not in terms of movement from one cell to another cell or one location inside the cell to another location inside the cell. The movement term here is with respect to the uh, presence of that particular genetic material in the whole genome. So, as we know that the word genome basically means all the chromosomes and all the genetic coding in all the chromosomes of that organism. Okay, so if suppose I'm saying that there is some XYZ gene and that XYZ gene is present on suppose chromosome 1. Okay, so now that presence of chromosome 1 from say location 1000 to 1200, that particular location is very precise and it is always present on that particular location only. Now suppose that if that particular gene needs to move from chromosome 1 to chromosome 2 for whatever reason. Okay, so it cannot directly migrate to that place. So what happens instead is that particular gene sequence from chromosome 1000 to 1200 location is copied in the form of RNA, and then RNA becomes mobile and goes to the chromosome 2 and basically gets converted back to the DNA, and that DNA is then pasted in the other location of the chromosome. So basically that is how the copy and paste mechanism, or that is how the movement in the genome by copy and paste mechanism is done. And this kind of mechanism, this genetic element which can do this entire thing, are called as retrotransposons. Now, of course, when we talk about anything that gets converted from DNA to RNA, the mechanism is called as transcription. But anything going back from RNA to DNA will be using the mechanism of reverse transcription. So, of course, when we are talking about any genetic material getting converted to RNA and then back to DNA, so that particular mechanism will be called as reverse transcription. And as I told you that we in case of retrotransposons, we are talking about the movement from location in one genome to the to some other location in that particular genome. So that means these particular elements will have the ability to integrate into the host genome. Otherwise, there is no point, because So it is getting copied from some place in the form of RNA. It is moving around, reaching the other place, getting converted into the DNA. But that DNA needs to be integrated into the genome. Otherwise, there is no point of that DNA. It will not get transcribed or translated or whatever function it is supposed to do. So that means it should have the ability to get converted, to get integrated in the host genome. So this is how a retrotransposon work. Uh, Talking about what impact they have on genome. So of course, if we are saying that any kind of gene is getting moved from one particular chromosome to another particular chromosome, that means that will definitely have some contribution to the evolution. Now there are multiple reasons why this needs to be done. There might be that some particular chromosome is getting shortened, or there might be a necessity for making multiple copies of that genes, and there can be multiple reasons why the organism decided to use retrotransposons, but ultimately that will contribute to the genome evolution. Then the second thing is it will affect the gene expression. Of course it will affect it. See, depending on where that particular gene or that gene element is getting integrated, it can either increase the expression. So suppose if that particular gene is, uh, integrating in a perfect location where it is getting transcribed and it is getting expressed, and it is the second copy of that gene, then in that case, the gene expression will increase. Okay. So in that case, the gene expression is going to increase. Now, um, the third thing is that they can cause damage or repair issues. Now, of course, if it is, suppose the genetic material has been copied and it got integrated in a perfect place under a good promoter and it is getting expressed, it is overexpressing, it is enhancing plants or any organism's ability, well and good. But if in case that particular integration happened in between some other gene, in between the coding sequence of some other gene, that means it is going to damage that DNA. That particular gene will either produce a misfolded protein, or that protein is going to be non-functional, or that protein is going to be of some other form, or anything like that can happen. So there are chances that retrotransposons can lead to DNA damage or repair issues. Fourth and most important thing is they can contribute to tumor progression. The same thing, the tumor progression is nothing but an outcome of DNA damage. And if we are saying that retrotransposons can damage the DNA, they will definitely at some or the other point lead to tumor progression. Uh, one thing we need to know is that there are two types of retrotransposons. One is long terminal repeat, that is LTR retrotransposon, and the other one is non-long terminal repeat, which is non-LTR. The reason I'm telling you the types of transposons is because the question that was asked, TOS 17, which the kind it is one of the kind of these two transposons. So basically, LTR retrotransposon, they are so the reason it is called as long terminal repeat is, so basically they are derived from some exogenous retrovirus infection. Some retroviruses use basically RNA as their genetic materials. So any kind of retrovirus infection can cause LTR retrotransposons. And in case of non-LTR retrotransposons, basically they are more primitive and ancients. They are very old kind of retrotransposons, which include long nuclear elements or short interspersed nuclear elements. They can be called as lines or signs, and these are the two types of retrotransposons. Coming back to our question, that is TOS 17. So TOS 17 is a kind of retrotransposon which is found in rice, and it can be activated by tissue culture or any other kind of stresses. So when we are saying that it is activated by tissue culture, that means one of the most important stages that forms in tissue culture is the callus formation. Okay, so these are some few listed features of TOS 17. Like TOS 17 is an LTR retrotransposon. That is the reason why I was explaining what LTR and non-LTR transposons are. That means this kind of transposon will definitely get activated due to some retroviral infection. And it is one of the few active transposons in plants, highly activated by tissue culture. It has low copy number and high frequency in transposition. It transposes randomly throughout the genome, and it is inactivated in regenerated plants. But despite all of these things, there are few applications of TOS17, like it can be used to create viviparous mutants in rice. It can be used to clone tag genes for forward genetics and can be used for insertional mutagenesis assays. These are the three applications where TOS17, a kind of retrotransposon, is used in different biotechnical tools. Also, it can be activated by some alien DNA interrogation. It is a non-autonomous LTR retrotransmission. That means it will lack any kind of sequence, any kind of GAG sequence. Okay, and it can be used to evaluate impact of transposition in rice genome. Since there are very few retrotransposons that are usually active, making good use of TOR17 was very important in case of rice genome, and TOR17 has done that work very well. So, coming back to our question, the way I told you that TOS17 will majorly get activated by tissue culture. So using this line, the question was that it is known to get activated at which developmental stage. Now, because we are talking about tissue culture stage, I told you that the most important stage in the tissue culture will be callus formation. So therefore, the answer to this question is callus. Okay, so TOS17 or retrotransposon is known to get activated at callus stage. Okay, I hope this question is clear. We'll move ahead to the next question. Okay, so which of the following techniques is used to detect the mRNA abundance of a particular gene to analyze its organ-specific differential expression pattern? If anybody knows the answer, you're most welcome to tell the answer. Okay, so now to answer this question, let's first understand the different types of blots. So here are the list of blots that you need to know. The first one is southern blot. So basically blotting techniques are nothing but they are used, there are gel based or some adherence based techniques which will help us understand the interactions between various things. So it can be the interaction between DNA samples or the interactions between RNA samples or between the protein samples or the either of the combination of these samples. So for the study of these kind of things, different types of blots are used. So the first blot is southern blot. Southern blot was basically the first kind of blotting technique, and the word southern here has been taken from the scientist who developed this technique. It is named after the name of the scientist basically who developed this technique. And southern blot was first used to analyze different DNA samples. Then later, once the southern blot was developed, people tried to develop the similar kind of technique for studying the RNA samples, and then it was named as northern blot. So southern blot was for DNA samples, northern blot was for RNA samples. Western blot technique basically uses the similar kind of technique to analyze proteins. So in this case, what happens is basically different types of proteins are transferred from the blotting sheets, and they are made to interact with different antibodies, and those antibodies are used to basically adhere and separate the different kind of proteins in the protein pool that we are providing it, and that is how the western blot is studied. Similarly, Eastern blots will analyze various post-translational modifications like ubiquitinylation or phosphorylation or things like this, which happens after translation to the protein. So the Eastern, so basically there are four main types of blotting techniques: Southern, Northern, Western, and Eastern. If you need to remember this, just try to remember the Southern blotting technique, which was the first technique, and you can the technique you can use to remember was anything that was first studied was our DNA. So the first technique also to study the DNA samples was Southern blot, which was the name of the scientist. The opposite for the Southern blot was named as Northern blot to study the RNA. Western blot was used to study the proteins, and the Eastern blot was used to study the post-translational modifications. Then there were various other techniques like dot plots, like far western plots, far eastern plots, and they tried to study different kinds of interactions between either DNA or RNA or various protein-protein interactions or interactions between glycolipids and things like this. So these were the major blotting techniques. And coming back to our question, that which of the following technique was used to detect the mRNA abundance? So mRNA abundance will be, if we move back to our this, we can southern blot was for DNA. mRNA is nothing but a type of RNA, so it will be northern blot. So the answer to this question is northern blot. That is, northern blot is the technique which is used to detect the mRNA abundance of any particular gene to analyze its organ-specific differential expression pattern. Okay. Now moving ahead to our next question. The following technique is employed for suppression of gene expression: X-rays, RNA interference, tDNA insertion, and promoter trapping. I'll give you 1 minute. If anybody knows the answer, please go ahead.
mRNA interference.
Yeah, correct. The answer is RNA interference. So basically, RNA interference is employed for suppression of gene expression. Now let's try to understand something about RNA interference. Um, Basically, RNA interference will just try to understand why does any organism require RNA interference. So what happens is that once the DNA is transcribed to form some kind of RNA, that RNA will get translated into the protein, and the protein is the one that is functional. Okay, so now if there is certain situation, say because of um, there is some kind of viral attack, or there is some kind of stress in the plant or in any kind of organism. So what that organism does is that it does not want that particular protein to get produced. It could be any reason. There are multiple reasons why this could happen. So the organism doesn't want that kind of protein to get produced. So what it will do is it will, but it cannot stop the transcription, because transcription is very tightly regulated, and um, to disrupt the transcription ways requires a lot of energy and lot of disruption in the control mechanism, which becomes very difficult. So what the organism does is it uses a side pathway to stop the protein production. That pathway is by disrupting the RNA that is produced. Okay. So if I try to explain you something in very short, what happens is we all of us know that DNA is a double-stranded genetic material, and RNA is the single-stranded genetic material. So being a single-stranded genetic material is the property of RNA. So once the RNA gets double-stranded, basically it becomes something which is not its original property, and the cell of that organism will recognize it as something which shouldn't have happened. So what it does is it initiates a mechanism where it will try to remove it. Okay. So what the organism, the cell of the organism does is that it will try to remove that double-stranded RNA because it is not something that should be present. And in case of in this process of removing that double-stranded RNA, what the cell does is that it activates an enzyme called as dicer. Now that dicer will do nothing but it will cut that double-stranded RNA into small fragments. And those small fragments are called as siRNAs or small interfering RNAs. Just give me 2 minutes. I think the students are still joining. Okay. Yeah. So what that dicer enzyme will do is it will cut those double-stranded RNAs into small interfering RNAs, which are called as siRNAs. Now, these siRNAs, so what happens is these siRNAs are also double-stranded. Okay, so that means that double-stranded RNAs will have some sequence which will be complementary to the mRNA that we want to basically whose protein we want to stop the production. So uh, that particular double-stranded RNA will get divided into single-stranded small RNAs, and one of the fragment of that small RNA, which is complementary to the mRNA, will get bound to the mRNA. And once that small interfering RNA gets bound to the mRNA sequence, it will cause the degradation of that mRNA. And once that mRNA is degraded, it will not get translated into the protein. So that is how the protein production is stopped, and this entire mechanism of stopping that protein production by degrading the mRNA is called as silencing of that gene. Now, why is it called as silencing of the gene? Because once the mRNA is degraded, basically the protein, even though we wanted to remove that protein, the function of that gene is getting stopped. Okay, so therefore this entire mechanism is called as silencing of the gene using the mRNA method. So this is how this method gets, working. We'll just understand this by this image. Yeah, like here is a good image. Let me just turn on the pointer. Okay, so see, this is the double-stranded RNA. It can be given in various forms. This can be externally given as well. If we want to deliberately stop this protein production or make a gene silenced or cause RNA interference, this dsRNA can be exogenously introduced. Or in most of the cases, the plant itself is able to produce this kind of dsRNA to protect the plant by RNA interference mechanism, also called as RNAi. So what happens is this double-stranded RNA will enter inside the cell and get broken down into small interfering RNAs or microRNAs. Small interfering RNAs and microRNAs are two different kinds of RNAs. The only difference is their size. Small interfering RNAs basically range from 20 to 25 nucleotides, whereas microRNAs can range up to 80 to 100 nucleotides. So depending on how dicer is cutting that, it will either produce small interfering RNAs or microRNAs. Now this cutting process will happen by the enzyme called dicer. And the adherence or the complementary siRNA sequence which will join to the mRNA is done using a complex called as RISC. RISC is a complex. It's a complex of different enzymes and different parts of the cells which will help uh, forming the bond between the complementary siRNA or miRNA with the mRNA that wants to be silenced. And once this RISC complex basically gets attached and this microRNA or small RNA gets attached with the mRNA, which is coming from the nucleus, this will get degraded and cut into small parts of the RNA and the functional protein will not form. Now let's talk about the application of this RNA interference. First of all, it can be used to treat the diseases. This happens in cases of basically humans or animals, that it can be used to treat hepatitis. And this is a very latest technique. So it can be used to treat hepatitis or different viral infections. Nowadays, it is also been used to treat cancers and various degenerative disorders and things like that. It can also be used for fighting antibiotic resistant bacteria. in a way in, which is very different to the way how antibiotics work. If you are interested in reading, there was also one of the vaccines for COVID-19 which was developed using RNAi techniques. If you are interested, you can go ahead and read about that. Then it is used to study various functional genomics where um, if you want to understand some kind of functions of some gene, you can it is basically called as loss of function phenotype. So if that particular gene is suppose responsible for producing pink color in a flower, and if you are silencing that gene, the flower should lose its pink color. So if you are silencing the gene and actually the flower is losing its color, that is called as loss of function phenotype, and that is how various functions of genes are also studied, which comes under functional genomics. RNAi can also be used in agriculture in various aspects. Some degenerative genes can be silenced, which are inhibiting the plant or harming the plant in some kind of ways. RNAi there is one latest technique where RNAi is used for basically deterring the pest which affects the different kinds of plants. That is another technique. So these kind of techniques come in handy when studying the agriculture, and RNAi has a lot of functions in that. So with this information, coming back to our question, the following technique for suppression of gene expression will be RNA interference. That is the only technique out of four which suppresses the expression by basically degrading the mRNA in the cell. So I hope this is clear about RNA interference, and we'll move ahead to our next question. Which of the following statement is correct for translational gene fusion construct? Subcellular localization of the protein can be disclosed. Protein trafficking can be traced and monitored. It has a promoter of gene fused with reporter only, and both 1 and 2. Okay, so we'll just move ahead to the answer. So for this, let's first understand what translational gene fusion construct is. So, a translational gene fusion construct is a genetic engineering design where a portion of the gene of interest is directly linked in frame with the reporter gene. So, if we say that any kind of gene is directly linked in frame with the reporter gene, let's first understand what a reporter gene is. So, if you're trying to understand something which is getting expressed, or a gene, if it is getting expressed, so reporter genes are basically those kind of genes whose some kind of physical trait is visible. For example, green fluorescent protein. Okay, so it's a kind of GFP protein. GFP protein, if the GFP protein gets expressed in any cell, that particular cell will get enlightened with the green color. Okay, so suppose there is a gene which is getting expressed, say in the midrib of the leaf. So if we tag that particular gene with a reporter gene, for example a GFP, so the midrib of that particular leaf should appear fluorescent green in color. Okay, so basically reporter gene is nothing, its function is to basically give a kind of visual phenotype which will help us understand or identify where all the gene is getting expressed or how much that gene is getting expressed. That is called as a reporter gene. There are various examples of reporter genes. For GFP is one of them. Luciferase gene is another. Luciferase gene is expressed in one of the insects, that the insect is called firefly. So it is expressed in that. That particular enzyme is also used as a reporter gene. So that is about reporter gene. So when we are saying that it is linked in frame with the reporter gene, so if we understand or if we just think about the basic concept of transcription, so transcription will start from a start codon, which will be regulated by a promoter sequence. Okay, so if there is a promoter sequence and immediately starts with any kind of start codon like ATG, that particular gene will keep on translating till the till it reaches a stop codon. Okay, so now if we try to insert any other gene which comes in the frame of start codon and stop codon, that means that means that it will form a protein in fusion with the protein of the reporter gene. Okay, so suppose there is a promoter sequence, and then we immediately added a start codon. Then we added a gene sequence of our interest. Suppose the XYZ gene. We added the sequence of the XYZ gene, and immediately without adding any kind of stop codon, we add the sequence of any reporter gene. For example, GFP in this case. Okay, so there is promoter sequence, there is a start codon, there is the gene sequence of gene of interest, and there is a gene sequence of the reporter gene, and then there is a stop codon. So what happens here is that we have linked a gene of interest in frame with the reporter gene. And now when the translation process will happen with this sequence, the translation process will start before the gene of interest and it will directly end after the reporter gene sequence. So there will be one single protein produced which will be called as a fusion protein, and that fusion protein will contain the sequence of the will basically contain the reporter gene protein also. So in this case it will also contain the GFP protein in along with the protein of our interest. That is why it is saying that translational process will seamlessly continue from the gene of interest to the reporter gene, and it will result in the production of a hybrid protein or a fusion protein with both the functions combined. That means that particular protein will also have the protein of our interest as well as the function of the reporter protein. Okay, so this will allow us to study the expression and the localization of the gene of interest by monitoring the reporter protein activity. So, as I told you before, that any kind of reporter gene or reporter protein, any protein will be termed as reporter when we are able to either see the expression or calculate the expression or calculate the monitor its activity or monitor its location or things like that. So once our interest of protein is linked with the reporter protein, we will be able to tell that we will be visually able to tell that how much that protein is expressed or where all that protein is expressed. And this mechanism of producing a fusion protein with in fusion with other gene of interest and a reporter protein, this technique is called as translational gene fusion construct. So if we try to understand this from this image, so if you look at this first image, this is a transcriptional gene fusion. And in this, what happens is that suppose this is a gene of interest and this is the reporter gene, and they both have their own promoter regions. Okay, so that means the gene will start its transcription from this promoter. It will take a start codon from here, produce a protein with the gene of interest, and then stop here. And then use another promoter, another start codon, take this reporter gene and stop here, and then produce another protein. Okay, so in this case, what happened is even though we have combined the two genes together, one after the other, the protein that is produced is separate because they have their own individual start codon and individual stop codon. On the other hand, if you look at the second image, which is called as translational gene fusion, in this case, what happens is we did not provide any other start site or any other promoter to the reporter gene. Instead, what we have done is we have one promoter, one start codon site, and the reporter, the gene of interest and the reporter genes are linked together. There is no space here. There is no stop codon in the middle of this. If there was a stop codon, the translational would have stopped here and the protein would have been produced separately as it produces in the first case. But in the second case, since there is no stop codon, the translational will simply end at the end of the reporter gene where the stop codon is present. And therefore, the protein that is produced here will be a fusion protein of gene of interest as well as of reporter gene. This will be a single protein and it will have the function of both reporter gene as well as the gene of interest. So this machinery is called as translational gene fusion construct. Now, what are the applications of translational gene fusion construct? It can be used to investigate various gene expression patterns, like it can visualize the reporter protein, and we can determine where and when and how much that gene is expressed within an organism. We can study the protein localization. Okay, that means by fusing the gene of interest to a reporter protein, we with a known cellular localization, we can track wherever that protein of interest is being distributed. And also, we can do the functional analysis of the protein domains by creating the fusion of different protein domains. We can understand the functional roles of very specific regions within the protein as well. So now moving back to our question, which of the following statement is correct for translational gene fusion? Subcellular localization of proteins can be disclosed. Yes, because once the protein is expressed, wherever the the monitoring protein or the reporter gene is being expressed, you can understand whether the protein is expressing in that particular location, and you can understand exactly where that protein is localized within the cell. The second is protein trafficking can be traced and monitored. Yes, wherever the protein is getting moved or however much there is a differentiation in the protein, if it is getting expressed more in some place, it's getting expressed less in some place, that all movement of the protein can be traced and monitored. It has a promoter of gene fused with reporter only. No, the promoter of the gene is not fused with the reporter only. Instead, the promoter of the gene is fused with both gene and the reporter. So the third option is wrong. First and second is correct. So the correct answer to this question will be both 1 and 2. I hope this question is clear. We'll move ahead to the next question. The reverse transcriptional polymerase chain reaction can be used to visualize spatial distribution of transcript in tissue section, quantify protein content in a tissue, study the chromatin structure, and detect the presence of transcript in a tissue. I'll give you 2 minutes. Please read the question, understand. And if you know the answer, please go ahead. Okay, so moving ahead to answer this question, let's first understand about reverse transcription polymerase chain reaction, or it is also called as RT-PCR. So what happens in RT-PCR is, if we break the words, polymerase chain reaction is nothing but a type of PCR, the normal PCR, where it is done in three steps, basically denaturation, annealing, and extension. That similar kind of steps happen in polymerase chain reaction. The only difference is that real-time PCR or reverse transcription PCR can understand the amount of transcription or the amount of transcript abundance that is present in your RNA that you have isolated, and it will basically use the reverse transcription mechanism or the reverse transcriptase enzyme to convert that particular RNA to the DNA, and then go ahead with the PCR reaction and then tell us whether how much amount of that particular RNA was present in our RNA pool. Now, if we try to understand it from this figure, so this was the RNA pool. So how this process is done is, first of all, we isolate the complete RNA content of any organism. So suppose if we are talking about plants, we will take a plant tissue and we will isolate the complete RNA from that plant tissue. Now that particular RNA, so the concept of real time or the reverse transcription RT-PCR is basically the amount of, so suppose you have a gene of interest, again let's take the gene as XYZ. So now the amount of gene, the amount of XYZ gene that is expressed will be directly proportional to the mRNA that was produced at that particular of time. Okay, so now suppose we have two tissues, we have taken two plants, and one plant was subjected to stress and the other plant was grown under control conditions. We isolated the same amount of leaves from both the plants and then took out the RNA. Now suppose XYZ gene, XYZ RNA was present more in case of stress plant and it was present less in case of the control plant. So now what do we infer from this? If any particular RNA is present more in any tissue, that means that gene was expressing more. Because when the transcription is happening more, that only then more and more RNA will be produced and more protein will be produced. So we basically do an inverse correlation here, that we are seeing that there is more amount of RNA present, and therefore we are correlating that there will be more amount of DNA, that is more amount of transcription would have happened. So when we take more amount, whatever the RNA we have isolated, we okay. So we are saying that reverse transcription is being headed by polymerase chain reaction, but we also know that polymerase chain reaction can only use DNA as its template. Fine. So, but here we have isolated RNA. Now, that means that we cannot use that RNA for a polymerase chain reaction. So we need to convert that RNA to DNA. And here the conversion of RNA to DNA will be called as reverse transcription by using the enzyme reverse transcriptase. And the DNA which is formed from the RNA will be called as complementary DNA. The reason it will be called as complementary DNA is the sequence will be exact complementary to the RNA without having any kind of introns. Why introns are not present? Because the RNA, the transcription process from the DNA to RNA would have removed all the intron sequences via post-transcriptional modifications. And therefore, the RNA that we are using will only and only have the exon sequences. And therefore, the exact complementary sequence of the RNA will form the DNA, which will be called as the cDNA. Now, this cDNA will be further used for the template of the PCR. Now, how are we correlating this? So, um, if you know the concept of polymerase chain reaction, if you give more initial template, your outcome. So, let's try to understand this with a very simple example. If you are doubling something, so there is one basket which has 10 fruits and the other basket has 2 fruits. So if you are doubling it every time, the 10, after 5 cycles, the basket with the 10 fruits would have more number of fruits as compared to the basket which had only 2 fruits. That means the initial amount of the template will matter to however much the product is being produced at the end of all the cycles in the polymerase chain reaction. So the more number of cDNA, the more the amount of product will be produced. And when more product will be produced, that means that if we are inferring that there is more number of cDNA and the cDNA was very much equal to the amount of RNA. So thus, if the product is more, the cDNA would have been more. That means the RNA would have been more. That means the gene expression would have been more. Okay, so that this goes in the reverse direction. Okay, so the reverse transcription polymerase chain reaction basically goes in the reverse direction. The more amount of product that you have, the more amount of the more amount of cDNA you had. That means the more amount of RNA you had. That means the more amount of gene that was expressed. So basically, RT-PCR is used to study the amount of gene expression or the transcript abundance in any cell. So if we are saying that XYZ gene is involved in suppose stress or it is getting more expressed in cell, that means the RNA that we isolated from two different plants should have the product from that stress plant should be more as compared to the control plant. And if the outcome after 35 cycles of polymerase chain reaction is more in stress plant, that means the stressed plant had more amount of cDNA substrate, which means it had more amount of RNA. That means the XYZ gene was more expressed in the stress plant, and therefore that's how we know the abundance, and that is the mechanism of reverse transcription polymerase chain reaction. RT-PCR, or the mechanism of this understanding of reverse, basically the real-time PCR, is done by the fluorescent dye called as cyber green, and the process is very similar to the polymerase chain reaction with the first step as denaturation, then primer annealing, and then extension. The only difference here is that when the extension is happening, cyber is a fluorescent dye. So every time a primer extends the DNA template, this cyber gets bound between the two strands of the DNA, or it is also called as intercalation. So the cyber basically gets bound to this DNA, and the more amount of product that will get produced at the end of every cycle, the more and more cyber will get bound, and the more and more fluorescence it will produce. And that is how the at the end of the RT-PCR cycle, we know that the more product is produced, and therefore we can go back to relating that the gene was expressed more. Uh, So in this case, there's one difference in case of producing the reverse transcription polymerase is when we are saying that the RNA is getting converted into DNA. The process is very similar to the polymerase chain reaction. That means it will require primers. So in case of conversion of from RNA to DNA, there are three different types of primers that can be used. One is called as oligo-DT primer. The other is called as random hexamers, and then one is sequence-specific primers. So sequence specific primers are nothing but whatever the sequence we are trying to understand. The primers are made from that particular sequence, and it will only amplify that particular gene in the entire mRNA pool. Okay, so the cDNA that will be formed will be only equivalent to the amount of the RNA that we are studying. Random hexamers are nothing but they are six randomly organized nucleotide base pair sequences which can get bound to any place in the mRNA, and they can start the extension from any place. And oligo-DT primers are just the multiple T nucleotides attached to each other. And as we know that mRNA, or the functional mRNA has a poly A tail, so it will have multiple sequences of A attached at the 3' side. So what this oligo-DT primers do is with multiple T nucleotides, it will get bound to the multiple poly A tail of the mRNA, and it will start the production of cDNA from that direction. So these are the three primers that are used for reverse transcription polymerase chain reaction. And with this information, coming back to our question, the reverse transcription polymerase chain reaction can be used to visualize the spatial distribution of transcript in a tissue section, quantify protein content in a tissue, study the chromatin structure, and detect the presence of transcript in tissue. So it cannot quantify protein content and it cannot do the chromatin structure. Visualize spatial distributions. Spatial distribution is a time dependent distribution. Reverse transcription PCR will not be able to tell the time dependent distribution. Therefore, the answer to this will be it will only help us detect the presence of transcript in a tissue. It cannot tell us that if there is any variation in the time dependence or whether with time the transcription is changing. RT-PCR cannot do that. So it can only tell us the presence of transcript in the tissue. So the answer to this question will be fourth option, which is detect the presence of transcript in tissue. Okay. I hope this is clear. We'll move to the next question. Okay, so the question is, which of the following techniques can be used to study protein DNA interaction? EMSA and yeast 1 hybrid assay, BIFC and chip assay, EMSA and yeast 2 hybrid assay, and yeast 1 hybrid assay and yeast 2 hybrid assay. You have 2 minutes. Please read the question and understand it. Let me know if you know the answer. Okay, so the answer to this question is the technique that is used to study the protein DNA interaction is EMSA and yeast 2 hybrid system. Now let's understand these two techniques. So EMSA is basically electron mobility shift assay. So how this is done is that a labeled probe or a DNA sequence basically A DNA sequence is mixed with a cell lysate, which contains different kinds of proteins that we need to study if we want to study some interaction between whether the that particular protein is getting attached with the DNA sequence that we are trying to study. So those two are mixed together and given time to form a reaction. So now if a protein of interest is actually binding with the labeled probe, then it will the reactions will happen where multiple things will generate. There will be some kind of, so for example here this circular protein is getting bound to the labeled probe. Then there is one this diamond shaped protein is getting bound to the labeled probe, and the triangle shape is not getting bound at all. Okay, so now this reaction we will run on the gel electrophoresis. Now what happens is there are chances that this protein that is getting bound will have more weight, or the protein size will be more as compared to the circular protein. So in that case, these two bands will separate on the basis of the weight or the size. So if the size will be more, it will take more time to move. So the bands will appear more in more upward direction as compared to the ones which are not bound. So when the DNA and the proteins are bound together, the molecular weight of that molecule will increase as compared to the DNA alone or the protein alone. So if you see on the gel electrophoresis or the imaging or via autoradiograph, you will see multiple bands here. So there, these bands could be the last bands will be of the free probe because they would have the smallest size and they will move the farthest. And here there are chances that there will be some proteins which show specific binding, or some proteins will show non-specific binding. And for so basically EMSA is a very basic technique, and there are high chances of non-specific binding in case of EMSA. So to make it more clear, there are other techniques like competitive or super shift EMSA, which is needed to specify specific bonding, but we are not studying that right now. So to understand EMSA, if there is any kind of protein which will get bound to the labeled probe or interact with the DNA that we are trying to study, can be visualized on the gel electrophoresis. And the other technique is East2 hybrid system. So in case of East2 hybrid system, the mechanism is that for any promoter to express any reporter gene, it requires a binding domain and an activating domain. So without binding domain and activating domain, the transcription will not take place. Until and unless these two domains have come close to each other, there will be no activation and no transcription will happen. So now let's understand this by four situations. In case of first situation, okay. So in first situation where our okay, so in case of this situation, where our gal4 DNA, this is a gal4 binding domain. Okay, so this binding domain will have a site for an activating domain. So once this activating domain gets in touch with the binding domain, this bit basically this activating domain will come closer to the binding domain, and then this binding domain will help in transcription of the reporter gene. So unless these two come together, the transcription will not happen. But in this case, since they are too close in close proximity and activating domain is bound to the binding domain, so the transcription will happen in this case. Okay, so then now let's take the second condition where what we did was we attached a bait protein to the binding domain. Okay, so this is the binding domain. So the binding domain can only bind to the promoter sequence. So unless there is a binding domain, nothing will bind to the promoter sequence. And we have attached the bait and prey here is nothing but the protein of our interest that we want to study whether they interact or not. Okay, so bait is the R protein of interest which is linked with the binding domain. And now we have not provided the activating domain. Now since there is no activating domain, no activating domain will come and bind with this, and therefore the transcription in this case will not happen. Okay, now let's take the third case where we only have a activating domain and we have attached a prey to it. Okay, now this activating domain cannot bind with the promoter sequence. And because there is no binding domain attached to this, the transcription in this case will not happen. Now, we have provided activating and binding domain. The these two domains will only come in close proximity if the prey and the bait will interact with each other. So once we know that if there are two proteins which are actually interacting with each other, bait and prey will come together, activating and binding domain will come together, and the transcription will happen. That means this LAC-Z gene will be expressed. But if these two proteins don't interact together, that means this binding will not happen, and in that case also the transcription will not happen because LAC-Z gene will not be transcribed. So this is how, by this mechanism, we know whether the two proteins that we have selected are actually interacting with each other or not. And this is how a yeast 2 hybrid system will work. And we are able to understand by this whether two proteins are getting interacted with each other. Okay. So I hope I am clear with this. If there is any doubt, please let me know, because it's already 8:15. If there's any doubt, we'll continue with that, or we just have one question remaining. We'll finish with that and end the session. Okay, this was the last question. So if there is any doubt, please let me know. If not, we can end the session. Okay, so I believe that there are no more questions and we can end this session.